Hello, my name is Joshua Mugabe. I welcome you to Chromosomes and Cell Division. A living cell seen under the microscope. It is often difficult to see the nucleus. So when you put a living cell into the microscope, you will see the structure like this, and it is not easily able to see the nucleus. When certain stains are applied, the nucleus takes up a stain more strongly than the cytoplasm does, making the nucleus easily visible. So when the nucleus or when the cell is stained, the nucleus takes up the stain more strongly than the cytoplasm. So this makes the nucleus easily visible. Stains can be chosen for particular parts of cell: the cytoplasm, the nucleus, the cell wall, or structures within the cytoplasm. So depending on the structure that is needed, or for observation or for studies, it will determine the particular stain that can be used. There are stains that are chosen for the cytoplasm. There are stains chosen for the nucleus, cell wall, and other structures within the cytoplasm. Staining usually kills the cell. So when the stain is carried out on the cell, it ends up killing the cell. By means of some special techniques of illumination, the nucleus and other structures may be seen in the living cell. So when we use different techniques of illumination, the special means techniques, those ones can help us to see the different structures of the cell, like for example the nucleus and then other structures that are in the cytoplasm. It is the difference in chemical composition between the nucleus and the cytoplasm that makes one take up a particular stain more strongly than another. So because of the different chemical composition of the cytoplasm, and then also the chemical composition of the nucleus, the stain that you use will determine, or the part that you want to see will determine the type of stain that you are to use. The living content of the nucleus is called nucleoplasm, to distinguish it from the cytoplasm. So we have inside the nucleus, that living content in the nucleus is what we call the nucleoplasm. It is distinguished from the cytoplasm, where the nucleus is embedded is called the cytoplasm. And what is found in the nucleus is called a nucleoplasm. In a cell which is not about to divide, the structures in the nucleus are not distinct. For example, if we look at our cell here, we have the nuclear membrane, which is surrounding the nucleus, and then we have the cytoplasm. But when the cell is not yet to divide, the structures in the nucleus are not distinct. They cannot be seen. So you can't see anything in the nucleus if it is not yet time for the cell to divide. Just before cell division, thread-like structures appear in the nucleus. Before the cell starts cell division stages, you will see thread-like structures in the nucleus. That is when the cell is about to divide. These structures are called chromosomes. So these thread-like structures that we are seeing in the cell, those structures, we call them the chromosomes. They get shorter and thicker and take up the stains very strongly. For example, when we are studying about the chromosomes and we have given the cell the stain, we shall see that these structures which are called the chromosomes, they are shorter and thicker and they take up the stain very strongly so that they are easily seen. The shortening and thickening continues. Now the chromosomes are seen in pairs. So you will start seeing the chromosomes, they become shorter and they become thicker, but you will see them in pairs. They will be in pairs. You will look at them and you will see them in pairs. They are distinguished by their size. You can identify them by their size. For example, this one, you know, it pairs up with this one according to size. This one pairs up with this one. Then this one is likely to pair up with this one. This one will pair this one, and this one. So they are distinguished by their size. The chromosomes are always in pairs because one of them is derived from the male parent and the other from the female parent. So when you look at them in pairs, this one and this one comes from the male parent and another one comes from the female parents. So you will see the same here. They are in pairs. One is from the male parent and another one is from the female parent. So that's why we're saying they are always in pair as they are derived from two parents. Different species have different numbers and shapes of chromosomes. So when you look at different species of organisms, you will see that they have different number of chromosomes and they are of different shapes. If you look at that one of the kangaroo, it has got 6 pairs of chromosomes. If you look at the one for CJs, it has 21 pairs of chromosomes and they are of different shapes if you look at them. If you look at the hawkweed, it has 4 pairs. You look at the chicken, it has 18 pairs. Look at the fruit fly, it has 4 pairs. Then human has 23 pairs of chromosomes, and they are different in numbers and also in shape. Members of the same species have identical sets of chromosomes. If you look at the members of the same species, they will have identical sets of chromosomes. For example, you look at the hawkweed, identical. You look at these ones of fruit fly, they are identical. That is if they are of the same species. Although chromosomes can be seen distinctly only at a time of cell division, they are present and active all the time. Though we saw that the cell before cell division, the chromosomes are not distinct, but the chromosomes are always present in the nucleus and they are always active. The chromosome carries DNA. The chromosome carries DNA. DNA is deoxyribonucleic acid. So this is our chromosome. The whole coiled structure is the one that we call the chromosome. And it is made up of a double helix. If you look at it, there are 2 thread-like structures moving around each other in a helical form, and they have a protein on which they rotate. They have a protein on which they rotate so that they cover shorter space, and the chromosome bears the DNA. DNA, this one controls all the chemical reactions in the cell. So it is the DNA that controls all the chemical reactions that take place within a cell. DNA also determines the species of organisms and its individual characteristics. So it is the DNA which determines the species, the particular species, that this is this species, and it is the one that determines the individual characteristics of that individual organism or that species. Chromosomes consist of DNA molecules supported by a scaffold of proteins. The diagram illustrates such arrangement, but it is really more complex than this. So when you look at our chromosome, the chromosome is made up of DNA molecules. And these DNA molecules, they are folded around a protein. They are folded around a protein so that they can cover a bigger area, or can occupy a shorter space. So, and it is also coiled. If you look at it, it is coiled so that it can occupy a shorter space. But it has also got the proteins over which the DNA folds itself. and arrange itself like that. But it looks like this, but it is more complex than the way it appears. The DNA in the chromosome carries genes. The DNA in the chromosome carries genes. So if we take this one to be our DNA, this DNA is on the chromosome and it carries genes. Or at particular points, there are genes. This is a chromosome, and this chromosome has got DNA and it carries particular genes. For example, here we have a single gene. Genes, here we have genes for tallness. Here we can have like genes for color. And here we might be having genes for eye color. This one is for hair color. So these genes are carried on the DNA, and the DNA is found on the chromosome. So those ones are referred to as genes. Consequently, the genes are spaced out along the chromosomes. So you can see that the genes are spaced out on the chromosomes. They are in different positions on the chromosome. That's where you are going to find the genes. The genes consist of distinct stretches of DNA. They consist of distinct stretches of DNA. Those are the genes. For example, here we have gene for tallness, and now here we have another gene for hair color, and here we have genes for eye color. So they consist of distinct stretches of DNA. By means of their DNA content, the genes control the activities of the cell, the type of cell it becomes, the species of the organism, and the individual characteristics of the organisms. So, depending on the content of the DNA, those genes there, they control the activities of the cell. For example, these genes here, they control the activity of the cell, and that is tallness of an organism. So they also controls how the organism is going to appear and its characteristics. For example, here you will see these are genes of eye color. These are genes of eye color. They are going to control the characteristics of the eyes, their color. The diagram illustrates the relationship between the chromosomes and the genes, but it does not represent an actual chromosome. So when you look here, it is a chromosome with the genes. You can see the particular genes for particular characteristics, tallness, a gene for hair color, genes for eye color, all located on the chromosome. The genes for these characteristics are not necessarily on the same chromosome, and the number of genes shown is arbitrary. So this is just an illustration chromosome, and it's not necessarily that the same chromosome contains the same genes. The genes for tallness is on the same chromosome as the genes for hair color or eye color, but it is just an arbitrary, just for us for illustration to understand what we are talking about. Because the chromosomes are in pairs, the genes they carry are also in pairs. We have said that the chromosomes are always moving in pairs. One of the chromosomes came from the male parent and the other chromosome came from the female parent. And because of that one, they also carry the genes. The genes are also in pair. For example, if we say this one came from the male parent and this chromosome came from the female parent, this gene capital A and gene small a, they are also in pair. They are on different chromosomes but located at the same point, so that when the homologous chromosomes come and align themselves, they are in pair. As you can see, B and small b, D and D, G and small g, I and I, they are in pairs. Each member of a pair of genes comes from either a male or the female parent, just as the chromosomes do. So you may find that if you look at these genes here, all these genes are on different chromosomes. This is a chromosome maybe from the parent, it came with this gene, and this is a chromosome from the female parent, it came with this gene. So these genes are located on different chromosomes, but they control the same activity. The individual genes of a pair, they control the same characteristics. For example, B capital and B small could control the eye color. Then G capital and G small could control the hair color. So what we are saying here is that these individual genes which are in pair, they control the same characteristics. For example, capital B and small b, these ones may be the ones controlling the eye color, and if you look at capital G and small g, these ones might be genes that are controlling hair color. Just before cell division, it can be seen that the chromosomes have replicated. That is, each chromosome has made a copy of itself, including its DNA. So what we are seeing here, we are seeing that the chromosomes have replicated. Replicated means that a chromosome now makes a copy of itself. That is when the cell is about to start cell division. So it is always one chromosome. It is always one chromosome. For example, it can be one chromosome like this. But when it is time for cell division, it will make a copy of itself and they will align themselves together. A copy like that. That is what we call replication. A chromosome replicates or makes a copy of itself, but they remain attached at one point, which we call the centromere. Now, these two sisters, we call them now chromatid. It is one chromosome, but now we call them sister chromatids. The copies and the original are also called chromatids. For example, here we have this one and this one, so we call them chromatids. We refer to them as chromatids. The copies and the original are called chromatids. So here we have our, let's say this was the original chromosome. If it was the original chromosome, it replicated, and after replicating, it made a copy of itself. When it makes a copy of itself, now the two are referred to as chromatids. The two are referred to as chromatids. Now the chromatids tend to separate but are held together by a special region called centromere. So they are held together at this point, which we call the centromere. The centromere appears as a constriction in the chromosome, but in a subsequent drawings, it is represented by a circle to make the position clear. So this point that we have here in the middle, where the two chromatids are held together, that constriction there is what we call the centromere. The chromosomes colored blue are derived from the male parent, and the chromosomes shown in red are from the female parent. So what we are going to look at now here, these ones that you see, the blue chromosomes, we shall take these ones to be the chromosomes that are coming from the male parent. And then the chromosomes colored red, we shall say these are chromosomes derived from the female parent. And this is a sister chromatid. You can see that point we said we shall now denote it by a knot in the center. That's where the chromatids are touching each other, or the chromosomes are holding themselves together. So that is the centromere. And these are sister chromatids, and these are sister chromatids here, and the sister chromatids. But we are saying that these ones that are in blue, these ones are from the male parent, and these ones are from the male parent. We have used only two pairs. We have used only two pairs, but the human being has got 23 pairs. But for clarity and easy understanding what is taking place during cell division, we are using only two pairs out of the 23 pairs. Two pairs of chromosomes. There are two pairs of chromosomes. Each chromosome has replicated to form chromatids. Now replication, this is a chromosome. It has divided itself into two to form chromatids. And this one was one chromosome. It replicated and it formed chromatids. The same applies to this one and this one. So we call them now sister chromatids, the two of them, one chromosome with chromatids. The nuclear membrane will disappear. That is the first part which is going to happen. The nuclear membrane, which was surrounding the chromosomes, disappeared. And when the nuclear membrane has disappeared, then we shall have the fibers appear in the cytoplasm, and they form what we call the spindle. These lines that we are seeing, they are the ones that we call the spindles. After the nuclear membrane has disappeared, then we have our spindles forming. Then the chromosomes, they move to the equator of the spindles. The chromosomes are going to align themselves at the equator, that is the middle part of the spindles. The spindle, but their middle part of the spindles is what we call the equator. So that's where the chromosomes will come and align themselves at the equator of the spindles. Although the shortening of the spindle fibers and the corresponding movement of the chromosomes are clearly associated, it should not be assumed that the fibers are physically tagging the chromosomes apart. So here we are seeing that when the chromosomes attach themselves at the equator of the spindles, the spindle fibers, they start to shorten and appear to pull the chromatids apart by their centromere. So when these ones, the spindle fibers starts shortening, we shall see that now the chromosomes will start to separate, and these are the chromatids now moving to different pole. This one was a chromatid, now it will be moving the other side, and this chromatid is also moving the other end. So the chromatids, now sister chromatids, will move to different poles as the spindles go on shortening. The chromatids are now chromosomes. Remember, this chromatid here was a sister chromatid to this one, and this one was a sister chromatid to this one. The same applied to this one, was a sister chromatid to this one, and this one was a sister chromatid to this one. But when they separated and they moved to different poles, moving to different sides, now they become each of the chromatid now becomes a chromosome. The chromosomes migrate to opposite ends of the cell as the spindle fibers shorten. So as the spindle fibers go on shortening, they go with the chromosomes, taking them to different or opposite poles. The cell now begins to divide. Now we are seeing that our chromosomes now have reached the end, and these ones have also reached the end. And now what's going to happen is that the cell now is going to start dividing itself into the middle. The cell now divides. We are seeing here the cell is dividing in the middle. So what is happening is that now the cell is going to constrict. Constricting in the center, it becomes smaller in the center, and it divides the other chromosomes that went up and then these chromosomes that went down. Now here, the chromosomes that went up, the nuclear membrane will form again and surrounds the chromosomes up, and then another nuclear membrane will be surrounded by surround these chromosomes that went down. So now the chromosomes will start becoming less distinct. They will start now disappearing. two cells are formed. Now here we have formed two cells. There is the cell on the upper side, and then we also have the cell on the lower side. So we have formed two cells. One cell divided itself and it has formed two cells. Now the chromosomes revert to their elongated thin shape and eventually cannot be seen. So the chromosomes that we have in the nucleus are going now to go back to their elongated shape, and they will become very thin, and now they will not be easily seen. Each cell now has a full set of chromosomes identical to the parent cell. So now this cell, they are going to become, now it was one, now they are two. And these two cells, they resemble the original parent. How the original parent was looking like, this is how these cells also look like. They have the same number of chromosomes, and they are also identical to their parent. In plant cells, the plant cells divide by building a new cell wall. Now for the plant cell, what happens is that the cell does not constrict and it divides into two. They just develop a new cell wall between the cytoplasm so that it separates the upper side and the lower side. We form a cell wall and it divides the cell into two. In animal cells, the animal cells divide by constriction of their cytoplasm. Now for the animal cell, what they do, the cell now will constrict in the middle until when it divides in the middle. So it will go on constricting until this end joins with the other end, and then they will become two cells. They will become two cells, one cell up and another cell down by constriction. So for the animal cells, for them they divide by the constriction of the cytoplasm, whereas for the plants, for them they form a new cell wall which divides the cell into two. Mitosis is a process of cell division which produces cells containing identical sets and numbers of chromosomes. So in mitosis, when the cell divides, we shall end up forming two cells, and those two cells that we form, each one will be identical with the old cell where they came from, having the same number of chromosomes and the identical chromosome. So this chromosome here, this cell here, will be identical to the original here. And the same applies to this one, will be identical to this one. Yet all of them are coming from the same cell. It is the same cell which divided and ended up forming two cells. But these cells are going to be identical in the set of numbers of chromosomes and also the type of chromosomes. Mitosis ensures that every cell of an organism carries identical set of genes. So in mitosis, mitosis is going to increase the number of cells. It results in increase in the number of cells. But now those number of cells are identical, meaning that they have the same type and number of genes in them, like the parent cell where they came from. So for it, every cell that is formed will be having identical genes, like the parent cell. Can you see a problem with this? Can you see a problem with mitosis? You see a problem with mitosis, whereby the cells, all the cells are produced, they have identical gene. Is there any problem that you can see? You can pause and then try to think about the problem which you think. The problem is that now every cell carries the same set of genes. How do cells then become specialized in shape and function to do different jobs? So the problem now that we are seeing here is that all the cells that are formed, they are identical. They have the same shape. They have also the same function. They have the same genes. Then the question is, then where will those cells come from, which are specialized in different shapes and different function? Yet it is giving us only identical cells with the same set of genes. For example, can a gene for brown eye do in a stomach cell? What can a gene for brown eyes do in a stomach cell? The answer is that the genes which are known to relevant to the cell's functions are not activated. So all these cells, they are identical. But when the cells find themselves in the stomach, it is only the genes that are relevant in the activities that are carried out in the stomach will be activated. Now, if the cells go into the eye, the cells will be the same as the cells in the stomach, but only that the genes in the eyes that are responsible, that are relevant for the activities of the cells of the eye will be the one that will be activated. Now, those ones that are responsible for the activities of the stomach, they will not be activated in the eye. We say genes for brown eyes is not expressed in the stomach cell. So that's what we are saying, that all the cells are the same. They are identical. Those ones you find in the eye, those ones you are going to find in the stomach. But if the cell is in the stomach, then the cells for the brown eye, the genes for the brown eyes in the stomach cells, it will not be expressed. Only the genes that are responsible for the activities that are taking place in the stomach cells are the ones that are going to be activated. In an onion root cell tube, a great many cells are dividing by mitosis, leading to rapid growth. So these are cells that have been got from an onion tip, root tip cell, and it is showing different stages of development of different cells. The preparations is made by softening the root tip tissue, squashing it on a microscopic slide, and staining the chromosomes and nuclei. So for us to get all this content, what we can see on a microscope, it was prepared by squashing it on the microscope slide and staining the chromosome and the nuclei. The squash technique, it spreads the cells out. See if you can identify the various stages of cell division. So you can look at these cells here and see if you can identify the various stages of cell division. You can pause and then you try to find out. Thank you very much for participating in chromosomes and cell division. Please do like, share, and subscribe.